Righto, hopefully while everyone's coming on now, we might just give it a couple of more minutes while we get a few people on. But welcome to the Double Down on Blackleg webinar. A couple of speakers will prepared for the day. I'm Chris Roberts from Nuseed. While people are coming on, so yeah, welcome and thank you for joining us. Righto. Just wait for a few more to come on. Anyway, we might kick away. So, yep, thanks everyone for joining us today. Myself, Adrian Carter, and Michael Hickey from Nuseeds will be hosting today's webinar. Nuseeds would like to thank Syngenta and the Seed Care team for all their help in hosting the webinar today with us. Yeah, wouldn't be able to do it without them. Righto. So, with major gene blackleg resistance in Australian canola varieties mostly overcome, it's important that we protect the genetic resistance that we still have. Just someone saying we need to maybe give it a couple of more minutes, but we just want to get on. We're on a bit of a time schedule here. So anyway, as people will come on, they'll just have to join us. So yeah, part of this is that, we want to protect this resistance. So a part of that is using highly effective fungicides on the seed or in the crop is a crucial tactic to keep ahead of this ever-evolving disease. So today, Nuseed and Syngenta are bringing together experts in canola breeding, fungicides, and blackleg pathology to discuss this in more detail and to share the latest information with you all. Some housekeeping today. So the webinar will be recorded and available to watch again and to share with any people that haven't been able to join us today. If you have any questions, please we'll have a question session at the end of all the presentations, or once all the presenters have spoken. Please pop any questions into the chat or question tool in Zoom, and we'll be able to talk to them or speak to them later on. Or if you're unable to do that, by all means, text them through to myself, Adrian or Mick. I've got their contact details up on the screen there for everyone. So I'll just give everyone a couple of minutes, a couple of seconds just to jot down those mobile numbers if you're having trouble with the question or chat function on Zoom. So hopefully that should be pretty straightforward. And if you already know Mick or Adrian or myself, you can just pop questions on the text message to us, and we'll be sort of watching our phones. So hopefully today we take away some useful information to help us protect our canola genetics and our fungicides which we're using, and which should in turn protect your canola yields into the future. So today we're going to hear from Nelson Gororo, who's Nuseed Australia's canola breeding lead. Sean Roberts from the Seed Care, from Syngenta Australia, so Seed Care Technical Service Lead here in Wagga. Pete Wilson, Technical Services Lead, Broadacre, New South Wales and Tasmania, also from Syngenta Australia. And Steve Marcroft, pathologist, Marcroft Grains Pathology. And then obviously we'll have the question session at the end. And then we'll close things out. Righto, no worries. So with further ado, I'd like to hand over to Nelson to have a chat to us today about major and quantitative resistance to blackleg in canola, and how Nuseeds is going about developing this in our germplasm. So over to you, Nelson. There you go, Nelson, you're there. Here it comes. What? Right.
Oh, good. All right. Thanks, Chris Roberts, and thanks everyone for giving us a chance to share with you what we're doing here at Nuseed. I'm based here at the Horsham Innovation Center. I'll just give it time to come through. All right, so I'm based here at the Horsham Innovation Center, and what we have is we've got lots of glasshouses, laboratories, and the work which we do is well supported by our team at West Sacramento in California, who do our molecular marker discovery work. And the molecular markers are tools which we use to speed the release of our Nuseed product portfolio. And recently, we have added the markers from markers for Blackleg disease resistance, thanks to GRDC Marcroft of grains pathology, the University of Melbourne. And our breeding process has several objectives. We do apply intensive pedigree and mass selection to identify lines with high yield, high levels of Blackleg disease resistance. We combine that with other key traits, agronomy, to improve grain yield and quality. I guess with us, we've got a good germplasm base, and the whole process starts in the glasshouse at Horsham. From here, all the newly inbred lines move to the disease nursery for at least 3 to 4 generations before we go to for yield evaluation. With each step, what we try to do is to condense the material down to a fraction of what we started with. One of Nuseed's key strength is having a good germplasm base. We've got a lot of germplasm derived from key breeding programs, namely the Exceed research, the NutriHealth program, DPI Victoria, Canola Alliance, and we also continue to source a lot of germplasm from other countries, spring types, winter types, semi-winter types from Europe, Canada, China, and Japan, with the objective to broaden our genetic base and enhance resistance to Blackleg disease. Through the use of our molecular markers, which have been developed at our center in West Sacramento, we can genotype our germplasm and come up with some clusters to identify lines which are similar, to identify lines which are dissimilar. And the diversity is strongly enhanced by geographic origin. In this particular case, material coming from Japan. It's enhanced by selection history, DPI material versus Canola Alliance. And we think this is a good decision support tool for our product portfolio development. Still continuing with molecular marker platform, this is just a quick summary of our Blackleg disease marker platform, which has been developed by the Marcroft grains pathology. What we have is material grouped into clusters or groupings, groups from Group A, Group B, Group C, right through to a rich cohort of germplasm coded here as unknown or yet to be determined. These are lines which are resistant to all known Blackleg resistance, and this whole process supports some of the uniqueness of our germplasm. The diversity provides a Nuseed with that agility to be able to respond quickly to pathogen changes. We are able to pyramid lines into varying major and minor quantitative gene combinations. I'll now invite Steve Marcroft to define what a minor gene is, what a major gene is. Thanks, Steve.
Thanks, Nelson. Just doing a bit of a tag team here in Horsham. I'll move on to my slides. So yep, I'm just going to run over today what your major genes are, your Blackleg resistance groups, and your Blackleg ratings, etc. So everyone's just a really good idea when we start talking about these genetics, what this actually means. So firstly, I just want to acknowledge that the work we do, or the work I'm presenting, is a national team effort. We've got people around Australia, and this is all funded by GRDC. Really, today I'm going to talk initially about this Blackleg resistance breeding, Blackleg resistance management, and then at the end of the day, or maybe not the day, the end of the session, talk about cultural practices and fungicide decision making, which obviously a lot of you guys will be interested in at the moment because it's that time of the year when we're deciding whether to spend their money on fungicides or not. So firstly, Blackleg resistance. We've got two different types of resistance in Australia, which I'm sure you're all aware of. We've got our major genes, which you'll know as the Blackleg resistance groups, which Nelson was just talking about. And when they're effective, they give complete control of Blackleg. So they cause immunity in the plants, which they're fantastic, but unfortunately they get overcome quite readily by the disease. And then we've got what we call quantitative resistance. And the quantitative resistance doesn't stop the Blackleg getting in, but it makes the plants quite resistant to the Blackleg. So it really slows down the development of the disease. So ideally with our quantitative resistance, by the time it gets to harvest, the Blackleg really hasn't progressed too far and it's not causing us too much yield loss. So in all Australian cultivars, they all have this combination of this major gene and this quantitative resistance in the same plants. I did talk to an eminent breeder once about what a good definition for quantitative resistance is, and his answer was it just means that we don't understand what on earth is going on, so we call it quantitative. But what is understood is that it's lots of minor genes added together with an additive effect. So if we go through to the next slide, and I'll click on the right thing. This is a bit of a, I guess, what you will see in your phenotype when you have effective major gene resistance. So with the effective major gene resistance, as I said, the plant is actually immune. You get no disease, and it doesn't matter what part of the plant. You get no disease on the seedlings, you get no crown cankers, you get no upper canopy Blackleg infection, no flower infection, etc., and no infection on the pods. The plants are completely immune. And the way this works in the plant is really simple. It's called a gene for gene interaction. It's where there's a gene in the plant which recognizes a particular gene or protein in the pathogen. And when the plant recognizes that protein, it turns on its defense mechanism and the plant is immune. The issue, I guess, is for the plant is that that protein in the pathogen is readily changed by the pathogen just through natural selection, mutation, etc. And when that mutation occurs, the plant doesn't recognize it, and you get this overcome effect here. And when the plant doesn't turn on its defense mechanism, the plant then becomes totally susceptible to those isolates. When these major genes are effective, you just get no disease. And it really doesn't matter where you grow canola or how you grow it, you always get no disease. So when we get one of these new major genes that the pathogen's never seen before, it doesn't matter what the disease pressure is, we can grow canola with no disease. Here's an example which I'm sure, well, anyone my age is aware of, this the Sylvester's resistance. So this is when we had this major gene resistance, and we got 3 years use out of it, and we got complete immunity in our crops, and everyone thought this was fantastic. People like me thought they were going to be out of a career, but 3 years later, that major gene was overcome, and the plants had no other quantitative resistance at that time. They only had that one gene and nothing else, and they completely died. So this is really, from a breeding perspective, one of the strategies we really try to avoid these days, so that it's all well and good to have this effective major gene in our crops, But what we want to do, and what the breeders like Nelson will do, is make sure we've got really good quantitative resistance behind it, so that when this major gene is broken down, let's say with a major gene, it's an R-rated Blackleg rating, one that is overcome by the pathogen, it just drops down to like an MR type resistance. It doesn't drop down to that complete susceptibility. And this is one of the really big changes which has happened over the last 20 years, sort of from a sustainable resistance management perspective, that we actually now have a much better understanding of the whole genetics of the Blackleg resistance, rather than at the time, all we knew was that Sylvestris was resistant and we didn't know how it worked or why it worked. It wasn't until the breakdown occurred that we started getting all this extra information. And look, from a breeding perspective, the industry really has moved on a very long way, and we haven't seen these issues really occur on a large scale since. So then moving on to our quantitative resistance or partial resistance, this is where we've got these little minor genes all added together. So if we've got an MS cultivar, there might be 5 of these minor genes. If we've got an MR, we might have 20 or 30. If we've got an R, it might be 30 or 40 genes. So we don't understand how this resistance works very well at all. GRDC is investing in a program at the moment to really try and quantify this and to give the breeders much better tools to be able to select for it. But really the process is, as Nelson showed in his 3 generations of disease nurseries, you'll just sow the plants in the field and you keep selecting each generation for plants look like this, and you assume that you're accumulating these minor genes. And that's really how it works. And then once you've got that, then you can combine it with a major gene, and hopefully you know that in your cultivar, you've got this great combination which will stand up to a whole range of Blackleg populations. So the big difference though with quantitative resistance is that it will change depending on the disease severity that you're growing under. I like to show MR as being an example. So we've got a good MR resistant cultivar. In an environment such as where I'm here in Horsham, where we're using a 1 in 4 year canola rotation, moderate rainfall, an MR is excellent. And in most years, you won't have any problems with it, won't need a fungicide. If you take that same cultivar into the Western District of Victoria, where you're growing a canola, wheat canola rotation, and it's really wet, that MR is still good, the plants won't die, but you'll still get this level of crown canker, and you'll then get a benefit from putting a fungicide, etc. on. However, if you're growing in a low rainfall environment, for instance, it won't change. So this, I guess, is where your scouting that comes in to see where your cultivar is sitting in each particular year. Now, quantitative resistance, we did hope 20 years ago that it was much more stable, but we now know that it actually does break down or erodes quite similarly to the major gene resistance. And in fact, I actually think it even goes at a similar speed. So this slide here, what I've done here, I've picked some nice, good Nuseed cultivars when they first came out and what their Blackleg ratings were. They were all rated as MR. So if you look at this in 2015, all our good Nuseed cultivars here are all MR resistance. And then when we look 3 years later, we can see they've ended up with something quite differently. And what this is this is the Blackleg. As we grow a good cultivar, we're selecting for individual Blackleg isolates that can attack that resistance, and we're excluding other isolates. So the thing with this is that the more successful canola cultivar is, the more Blackleg there is that can attack it, and the more we overcome it. So it's unfortunate, but the only way we can really protect our resistance is by not growing our cultivars. So from a breeder's perspective, they've got to grow cultivars which nobody wants to grow, which obviously isn't a very good business strategy. But if you grow cultivars which are high yielding and whatever, and you grow them year after year, what you do is you select for Blackleg isolates that can overcome that resistance. And we see this quantitative resistance being overcome. And we know that it's different in different cultivars because they will do different things. So like you said here in this slide, they all started up as MRs. They all look exactly the same. 3 years later, we can see that some cultivars are looking quite different to other ones. So just finishing up this session now, in Australia, obviously the first thing you look at is the Blackleg rating of your cultivar. And the way we generate the Blackleg ratings is in these disease nurseries. Disease nursery is simply growing canola on last year's canola stubble. We sow all the cultivars and all the breeders' codes, etc., all the new germplasm in rows, and we count how many plants germinate for every single row. And then we come back at the end of the year and we count how many are surviving. So you can see here, there's one here, they've all fallen over and died from disease. So that might be an MS type cultivar. And this one here where they've all stood up, might come out as an R-rated cultivar. So it's really just a survival percentage. We have 8 of these disease nurseries across Australia, so we get lots of diversity in all of the canola growing states, and they're all funded by the GRDC. The other part of the process with the Blackleg resistance groups, how we do that, as I said before, it's a gene for gene interaction, and you can see here the effects we get. So we'll put the same isolate on this line, on this line, on this line, on this line, and you can see here, this cultivar contains the gene, so it holds the disease out and you get no disease. This gene, this cultivar here didn't have the gene, so it gets invaded and you get this lesion development. And through this process of patterns, we've got 16 of these different isolates which can attack individual genes. We can then identify which genes are in which cultivar. And look, this, as Nelson said, this is now at the moment being converted to molecular markers, so it's going to become much more streamlined. But this work's all been developed in Horsham and it's used right around the world now. So I think that is where we hand over to Syngenta, but hopefully that gives you a good understanding, or sorry, back to Nelson, and good understanding of the quantitative and major gene resistance.
Thanks, Steve. I think it's a nice segue into my presentation here. And just to answer the question is, where does Nuseed do its testing for Blackleg resistance? We do most of our work at Laharam, which is right on the foothills of the Grampians National Park. It's a cool, wetter environment, plus 400 mils annual rainfall on average, and it's ideal for screening of Blackleg disease, and it's also close to the R&D base. And we also have a good relationship with the grower. So every year we do run 2 disease nurseries, and in 2020, our main nursery, which is really our breeding non-GM nursery, was on an ATR stingray stubble, and the commercial site was on a Bonito stubble. So the commercial site houses all our material, which is in yield trials, advanced lines, demo lines, and some of the industry projects which we do. And the success of a good disease nursery depends on if it's a function of the environment, and Steve has already outlined that. In particular, Laharam is pretty good in the sense that, you know, you get 3 weeks of continuous damp conditions, and we get temperatures less than 15 degrees Celsius. And we're also talking about the genotype of the varieties which we test. We've already demonstrated the good germplasm base of Nuseed varieties. And lastly, you know, the currency of a good nursery is the pathogen type. And it's not unusual to have 10 to 15 strains of fungus at Laharam. I guess with our strategy, you know, it helps us to screen out the worst from the best. We fix lines from F3 to F7 generations. We encounter adverse growing conditions. We bag lines to encourage self-pollination and to prevent outcrossing, and lines are screened for Blackleg disease. So in this particular slide, you get susceptible lines here and the resistant lines. And the first thing which we do is also evaluate our material for seedling resistance to Blackleg disease by looking at visual leaf lesions, where a score of 0 is represents no visual lesions and 4 is dead. And we're also looking at the time, or the amount of time it takes for seedling to die under extreme disease pressure. The last one Steve was already touched on, which is really looking at the adult plant resistance assessment. I'll talk about one of the approaches which we use in screening double haploids in a humidity chamber. Nuseed produce on average 6,000 double haploids per annum, and they are becoming the dominant form of new lines in the pipeline. And double haploidy is a tool which we use in a tissue culture lab, where we start off with just a single pollen grain, and we double the chromosome number of that single pollen grain. We produce plants, and these plants are pure, they are fixed. And the beauty about this process is the time to develop a varieties is shortened by 2 to 3 generations. How does the process work? What we have here is plants or seedlings at 1 to 2 leaf stages are transferred into a controlled environment, which is where you've got canola stubble, which acts as a source of the inoculum. They stay in this chamber at high humidity for 24 or 48 hours, and then they are transplanted into big pots, assessed for seedling and or adult plant resistance. And we expect to lose at least 30–40% of the DH, which means the less material go out in the field, which means only the elite material end up being in the field. So in closing, Nuseed have got a significant level of quantitative resistance to blackleg disease. We've got a considerable range of germplasm diversity. We've got a good team, which is experienced and dedicated. And we have good associations with organizations such as the Marcroft Grains Pathology, NBGIP, which is funded by GRDC, universities, and Syngenta. Thank you.
Thanks, Nelson, Steve, for that rundown on plant genetics and their blackleg resistance. Next, we'd like to welcome Sean Roberts for the next presentation. Sean is the Seed Care Technical Services Lead for Syngenta Australia and leads the Seed Care Institute based at Charles Sturt Uni in Wagga Wagga, which focuses on seed application testing and training, supporting Syngenta's leadership in seed care. Sean will provide an update on the Sultra Duo trial results, blackleg and Pythium resistance management. Thanks, Sean.
Thanks, Adrian. Do you need my video? Can everyone hear me or Nelson?
Yes, we can hear you, Sean.
No problems. All right, thanks very much for your time today. Today we're just going to walk through, look at the control of seedling diseases in canola and look at the group 7 SDHIs, which is in Sultra Duo versus the DMIs. I can't start it because of the host. So versus the DMIs. So just introducing Sultra Duo and what it is. It's a mixture of 2 products, which comes in a co-pack. First product is Sultra, pididiflumethofen, and the second product, Maxim Excel, with fludioxonil and metalaxyl M for the control of dampening off diseases. It's an FS formulation, red in colour, but the registration is canola, and the diseases that we do have is for the control of seedling blackleg, dampening off, and root rot caused by the complex of Pythium species and Rhizoctonia salii. The use rate is 400 mils, 200 of each mils of each product, adding up to that 400 mils as a seed applied fungicide. How does Sultra work within the plant and provides that protection for the cotyledons and early leaves from blackleg infection? Is Sultra is a systemic seed treatment. So the images below we have here radioactive isomers with the active ingredient, and it's applied to the seed. And we can see the green here and the blue. So the green is where it's applied to the seed, and you can see after that the product moves out into the soil and up through the roots via the xylem, and it's moved through the plant systemically. It also has some uptake during imbibition as well at germination. So you can see here that early protection is provided by the active ingredient in the cotyledons, and we see a good concentration of the product at this most
stage and most vulnerability for infection here that leads to stem cankering at the end of the season. We see that again at 14 days, we see it starting to move into the first true leaf and the second true leaf. And as we get to the fourth leaf stage here, we can see whole plant protection there with the active ingredient, the plant there, from infection from blackleg. We move here to the 28 days. We can still see the cotyledons have dropped off by this stage. You can still see the first true leaves have good amount of active ingredient. And you can see just now the sixth leaf has a small amount of active ingredient there. And beyond that, we don't see active ingredient there within the plant. So this is really important. It comes back to when we start talking with resistance management as well, when we start following the crop life procedures. So again, the systemic active ingredient being systemic gives that whole plant protection at the beginning of the season and helps deliver that improvement in disease control. If we come back and just look at the SDHIs and Saltro Duo being a new mode of action in canola for a seed treatment, it is an SDHI, part of our SDHI family within Syngenta, but work that has been done, survey work that has been done through Marcroft pathology and the University of Melbourne, and work that's also been funded by Syngenta to come back and look at a baseline to see what resistance is out there in the field, or if there is any resistance with the SDHIs. From 2018 to 2020, survey work of over 380 paddocks were found, and we had found that there was tolerance out there to fluquinconazole, which is the seed-applied DMI, such as Jockey Stay or Jockey, and back to flutriafol with products used in furrow or on fertilizer there. So the mid-20% tolerance and up to the high 20% intolerances with the flutriafol on fertilizer. The good take-home points away from the work that was done by Steve and Ange is there was no known resistance to SDHIs or Saltro in canola, and also the SDHIs or Saltro and Miravis, there's no cross resistance with the DMIs. Again, just a quick picture of just highlighting this from some of the work that Steve and Ange have done. It's a cotyledon disease test with infection there. We see the Saltro of healthy cotyledons. The untreated here with no cotyledons alive. We have a one DMI who had some tolerance, and over here with a DMI that didn't have any tolerance there. So it's a really good baseline for us to move forward into the future and see if there's any shifts in the population with the SDHIs. Well, Steve will have a little bit more later. As we flow back into the resistance management, the first point I want to make is, come back and follow the Crop Life Australia resistance management strategies for the fungicides 2020 to 2021 guidelines there. But come back in, following up some extra points there is we want to be making the industry needs to be following the GRDC blackleg management strategies out there in the field with cultural practices and the use of varieties with resistance. Also come back and look at using fungicides as preventatively, and also come back and using numbers and modes of actions to help give that resistance management piece there as well. If we come back and we start looking at using a group 7 seed treatment at the beginning of the season, and with Saltro, if we want to come back and do a seedling spray there at that second, fourth leaf spray or the sixth leaf spray, then that first spray must not contain a group 7 fungicide. If we come back into later the season, then we can come back in at that 20% flowering for that upper disease management for the canopy there. We can come back and look at using a group 7 fungicide there. So just to hit home those 2 points down the bottom, if a group 7 seed treatment is being used, the next fungicide, if we come back into control blackleg, has to be a non-group 7 fungicide. And also during the season, we do not apply more than 2 applications containing a group 7 fungicide for the growing season targeting blackleg. So hopefully that's kept it nice and simple there. But if you do need to chase up further information, you need to read up more on, there's more information there at Crop Life. If we come back, I'm just going to talk through some trial results here today. and just looking at the SDHIs and the DMIs there. But when we're looking at out there in the field, this is for stem cankering at the end of the season. This is the most critical stage with the canola crop, and this is where we're looking at the seed treatments to provide protection from the healthy cotyledons through to that 4th to 6th leaf stage there. And the photograph here comes from our learning centres at Wagga Wagga with Syngenta. The bit of trial work that I'm going to bring through is we're going to be looking at healthy cotyledons. This is across 30 trials across WA, South Australia, Victoria, New South Wales, done by internally Syngenta field biologists and independent research organisations such as Marcroft pathology. And what we're coming to here is looking at the bigger the number, the better. And we're looking at just either have we got a healthy or unhealthy cotyledon, so with or without disease. So all the trial work we've done here does have good disease pressure. We're coming back and looking at untreated here, only having 20% healthy cotyledons. With the SDHI technology here in Saltro Duo, we're looking at that has been lifted up significantly above it to about 75. Back at looking at the jockey here with the DMI technology, we're looking at around that 55% healthy cotyledons. So it's a significant difference to the Saltro Duo. What we found from all the trial work that we have pulled together here, 84% of trials were significant over the untreated and 50% over the jockey stayer were significant in improved disease control. As this flows on to severity, internal infection at maturity, again, we come back and look at this, the smaller the number, the better, with better disease control. And we come back and look at the untreated here with about 55% severity, the Saltro Duo around that 30% severity, so seeing a significant reduction there of 97% of trials versus the untreated. And when we come back and look at the jockey stayer, about that 45% severity, and back compared to the Saltro Duo, about 60% of the trials will see a significant decrease in disease pressure. Again, these trials were done across exactly the same locations as before. And coming back and looking at yield and what does it mean with yield here, we're looking at two different varieties, an MS variety and MR variety. We're both seeing positive effects with yield from the DMIs back to the Saltro Duo there. Again, just looking at the common dampening off there, it is commonly across Australia for Predictor B showing Pythium to be everywhere and Rhizoctonia as well. Saltro Duo provides protection against dampening off, increasing root growth there, protection and more uniform seedlings there, and we're not missing any dampening off of seedlings there. With our trial work, when we're developing Maxim XL, 15% improvement was seen compared to the untreated when we saw the disease of Pythium out there in the field, and this was highly significant with a very low p-value. And coming back and looking at Maxim XL, again there with Rhizoctonia trials, we saw an increase with 13% with plant numbers there with Rhizoctonia with a low p-value and highly significant as well. Thanks very much for your time. If you have any further questions, ask them at the end or come back to myself and I'll pass on to Pete Wilson.
Thanks for that, Sean. Up next, we do have Pete Wilson, who is the Technical Service Lead for Broadacre, based out of New South Wales and Tasmania. Pete has been focusing on the fungicide portfolio for Syngenta for the last couple of years, and in particular, the launch of Marava Star. Today, he's going to give us an update on Marava Star for the use in canola, mainly around Blackleg, both seedling and upper canopy management. Prior to being with Syngenta, Pete does have a wealth of knowledge. He's been an agronomist for 30 plus years. So if anyone does have any curly questions in regards to disease management, please put them in the Q&A and we will endeavour to get to them at the tail end of this session. Over to you, Pete.
Well, thanks, Michael, and thank you everyone for your time today. So this afternoon I'm going to introduce Marava Star. So it's a new foliar fungicide from Syngenta, where we're looking at controlling the key diseases in both pulses and canola. At this stage, registration is expected for Q3 of 2021. So over the last couple of years, and particularly last year, given COVID, etc., certainly Marava Star was showcased at our learning centres across Eastern and Southern Australia we had trial sites in at Wagga, Horsham, Roseworthy particularly, where advisors and agronomists were able to see Maravis in those early stages of our development process. In addition to that, we also had Maravis Star trialled extensively last season under a large-scale permit as well. And we've also had strong industry collaboration with the various government research bodies over the last 2 to 3 years, including Marcroft grains as well. So what is Maravis Star? So it's a co-formulation of pitoflumethafen and fludioxonil, and the co-formulation will be brought to the market to give us a dual mode of action for our resistance management going forward. And so the pitoflumethafen is the Group 7 or the SDHI component, and the fludioxonil is the new mode of action, the phenylpyrrole Group 12. So registration at this stage will be in canola and pulse crops, and we'll have a rate range of 600 mils up to 1,000 mils per hectare, with various disease criteria, control rates subject to the label being finally approved. Diseases controlled at this stage, canola looking at 4 key canola diseases and 5 key pulse diseases. So if we look at canola specifically today, the purpose of the webinar, so we're seeing very good activity on seedling blackleg at the early stages, at that 4 to 6 leaf stage. Also controlling white leaf spot as well, which we saw excellent results last season, given the presence of adequate moisture from early on in the season. And most importantly as well, seeing excellent control of upper canopy blackleg and also sclerotinia when applied at that early flowering stage. So how does Maravis Star actually work? So bearing in mind that we do actually contain two active ingredients, pitoflumethafen and fludioxonil. So firstly, talk about pitoflumethafen. It's very stable active ingredient where it doesn't show any volatility. It's extremely stable when it's applied to the leaf surface. It has a very strong affinity to the waxy layer. And probably one of the other key attributes of pitoflumethafen as well is that it's extremely rain fast. So if you actually look at the diagram here, you can see that when pitoflumethafen is actually applied onto the leaf surface, we see very rapid uptake into the waxy layer, and it basically occurs within the first 6 hours after application. And then what happens then is that actually pitoflumethafen, it is lipophilic, so therefore it has a very strong affiliation for actual wax. So it locks itself into that waxy layer, and that provides the long-lasting residual from this particular active ingredient. And over time, you'll see the slow movement of the pitoflumethafen into the leaf tissue, and then you'll see limited movement further through the xylem over time. So in terms of fludioxonil, so the characteristics of fludioxonil is that once again, it is a highly residual active ingredient which binds on the leaf surface. It's extremely rain fast, and most importantly, it lowers our resistance risk for the fact that we are actually introducing a alternative mode of action. So in terms of this is the activity of fludioxonil. It works on the leaf surface. Okay, so as you can see there, it basically had three particular areas or modes of action. It stops the spores germinating, it inhibits the growth of the mycelia, and it prevents these cells from undertaking normal. So if we look at the application guidelines of Maravis Star for canola, as Sean has alluded to previously, we'll be looking at two specific timings as part of our crop life resistance strategy in terms of our blackleg management. So selection would be for non-SDHI seed dressing is not applied at planting, and you do have early infection of seedling blackleg. Maravis Star will be a suitable fungicide at 4 to 6 leaf stage. However, if a SDHI seed dressing has been applied, you would then look at that timing later in the crop, which coincides for upper canopy blackleg is from stem elongation through to 20% flower, and sclerotinia not later than 20%. Part of our crop life resistance strategy will be limited to one application per crop, once the crop has emerged, that is. Adjuvant will not be required, and we will have registration for both aerial and also ground application. And the rainfast will be 1 hour. Just reinforcing the application guidelines here, just giving you the either or option in the actual crop, either at the 6-leaf stage or at that early flowering, depending on your expected disease. So field experiences from last year in 2020, what did we see as part of our large-scale replicated trial program? There's some work which was conducted where we planted a susceptible variety. It's 21 days after application here, looking at seedling blackleg. So just showing you the degree of lesions there on the lower leaves. And what we saw was significant dose rate response with Marcroft Star. Lower rates, 400 mils, working through to our label rate, bottom label rate of 600 mils, showing excellent control of seedling blackleg. The important thing as well we saw last year was just that step up in blackleg control of an SDHI versus existing DMI chemistry. Very visual. The other thing we saw as well is upper canopy blackleg, excellent control later into the season with the extended residual of Marcroft Star, and we saw a green pods, and certainly there was a yield advantage. The other side effect, apart from upper canopy blacklegs, also control of sclerotinia, and very noticeable last year, and that has been replicated over a number of seasons at this point in time. So the main thing to point out with the sclerotinia control as well as the upper canopy blackleg is the extended green leaf and the actual retention of those green leaves. Just rounding it out, just some trial results from last year, just showing you with all SDHI fungicides, certainly are seeing a yield advantage and compared to the untreated. So with that, I thank you for your time today, and any further questions, by all means, refer them back to us over time. Thank you.
Adrian, we can't hear you.
Okay.
Thanks, Pete. Sorry about that. We have heard from him already today, but we'll get him up again to wrap the session up. Steve Marcroft needs little introduction to most people in the ag industry, having been working on canola diseases for the past 25 years. Over his career, Steve has been instrumental in the development of many of the cultural practices currently used in Australia to minimise the impact of blackleg on canola crops. Steve's going to give us the update on current industry research and bring together the practices to help minimise the impact of blackleg on canola. Thanks, Steve.
Thank you. Now we just need to share that. Share screen. Is screen one?
No, two, sorry. Two.
Okay, hopefully we're away. Nope, we're not moving. All right, now we've skipped forward. No, that's right, we're away. Great. So yeah, I'm just going to run through now sort of blackleg management and then spend a bit of time on decision making with our fungicides, which obviously this time of the year, a lot of people are thinking about, depending on whether they get their crops in or not and whether they got rain or not. So just to start with, because I think this does help you understand why we do what we do when we manage blackleg, just quickly run through the blackleg life cycle. So one of the key things with blackleg is it survives on canola stubble, and that's really significant because it means that you grow a lot of canola last year. It means you have a lot of inoculum this year. It means you can't get away with it. It's not like a rust on a wheat where it's an obligate, where it has to live on green tissue and it dies down each summer and then starts off. So you have those years where you have the green bridge is more severe. With blackleg, it's severe every single year. And it really means if you're in an area that grows, canola, wheat canola, you've got that really high level of nitrogen straight away. The other key point is that it's sexually reproducing. So all these little fruiting bodies on the stubble, which all throw out these spores every time it rains. And, this week we drew a bit of rain and I expect lots of spores and lesions to be turning up from that. They're all genetically different to each other. So they're all individuals because they're a result of sexual reproduction. And that means we have this amazing genetic diversity within the crop, which is why we overcome our resistance so quickly. So when you grow a brand new resistant cultivar that Nelson's grown, produced for you, the moment you grow it, you start for selecting these individual isolates which can attack it, and then year after year, they build up in frequency and they overcome that resistance. And then you have to go and switch to the next brand new canola cultivar. So these spores land on, they cause a seedling infection. And from a crown canker perspective, this is really the most important stage because it's the cotyledons and the first couple of leaves that are infected, the pathogen then grows down the vascular tissue down to the crown, which causes that crown canker damage. So, which is why we're talking, like Sean was talking about the Saltro, it's really, from a crown canker perspective, it's really about protecting that seedling. Once you protect that seedling and get it past the 5th leaf stage, any infection after that, sort of this type of infection doesn't really matter anymore. It's really superficial because it's too late to cause that crown canker, which we see at the end of the year. So that's how it works. So what's the first thing? The first thing is to understand your risk. So, and I think this is actually really important and gets forgotten about sometimes. Like if you're in this photo series taken on Lower Eyre Peninsula, where they like to grow canola, but in a canola wheat canola rotation like this, when you grow your canola, you just can't get away from last year's stubble. You're going to have stubble paddock, stubble on 2 sides of your canola paddock from last year. So you've got this enormous amount of inoculum. So you combine a canola wheat canola rotation with high rainfall and you're going to have blackleg problems. But the point I want to make is that a lot of Australia, we don't grow canola quite so intensively. Like for instance, if you're here in Horsham in the Wimmera, you're probably much more likely to be a 1 in 4 year rotation. And you can then avoid this canola stubble and you don't have the disease pressure and then you don't have the evolution of the pathogen overcoming resistance genes and etc. So it is important to understand, are you in that really high risk situation and you have to do everything you can to avoid blackleg, or are you in an environment that's not anywhere near as so aggressive, I guess, and you can keep cultivars for a lot longer. So the second step is really in reflection of that environment, how much diseases have actually got. And that's why I really encourage everyone, straight after windrowing, when it's easy to walk through the crops, to get out there and cut plants and have a look. Have I got dead and dying plants? And if I cut them over my secateurs, if my plants look like this top row here, that's great. Continue with what you're doing. You already don't have many problems. If the plants look like the bottom row, that's when you're getting yield loss and that's when you need to change your practices in future years. And the other thing to remember is with blackleg, you can tolerate a little bit of disease and not actually get very much yield loss at all. Because if you think of this as like a partially blocked pipe, as long as you've got enough vascular tissue, the plants can still survive and do quite well. It's when you get this level of disease that it gets too critical and you start getting that yield loss. The next stage is the cultivar resistance. Go to the blackleg ratings. That's the most important thing you can do after you've avoided canola stubble, etc. And I think it's worthwhile saying just how successful the blackleg ratings have been. If we go back 30 years ago, we couldn't grow canola because it just died from blackleg, and now we can produce over 4 million tonnes, year in, year out, without getting a lot of yield loss. So it's been an incredible success story. And I don't know what the percentage would be, but I'd say 70 or 80% of that success is due to this genetic resistance, due to this breeding. Everything else that we do is really trying to support the resistance. But it's about the breeding, which has really developed the industry. And the other thing is, as we've been talking about, the pathogen will overcome the resistance in our cultivars. We know that it'll also overcome our fungicides. So it's really critical that you actually do look at the blackleg ratings each year, because the blackleg ratings will change. We know those cultivars will erode in resistance. We know major genes will get overcome, which is why, generally speaking, if you get onto a really good cultivar and you really like it, you get around that 3 years use out of it, and then you'll start losing yield again to blackleg. And then it's about selecting another cultivar from the blackleg ratings, which has got really good resistance. And there's another reason why go out and check how much disease you've got in your crops each year, so that you'll know whether that resistance is being overcome or not. Next stage, step 4, is trying to get a distance between this year's canola crop and last year's canola stubble. This is some photo I did a number of years ago now. Yeah, in my PhD over 20 years ago, where we really found that 500-metre isolation from this year's crop and last year's canola stubble was about the best you could get to reduce the amount of disease. And that's still very applicable in most canola growing regions in Australia. Obviously, in our canola, wheat canola, that's been thrown out and it's really been replaced with the fungicides to stop the disease in those regions. Talk about resistance management. And this is really where the knowledge of the genetics of all our cultivars comes into play. So in the past, when we started losing resistance, we could potentially switch cultivars, but then grow cultivars with very, very similar resistance and not get a lot of benefit. Whereas now we know what the resistance genes are in our cultivars. That's the Blackleg group. So we know whether it's group A, B, C, or it's a combination in A, B, C, which simply means it's got 3 of those major genes combined. We know that if you're losing resistance in one cultivar, you can look at the resistance groups and then change to a cultivar based on a different resistance group. And as Nelson said, that's why the breeders these days do put effort into breeding cultivars based on those different resistance genes, so that growers have got options and you're not just always selecting against the same resistance gene. So that's good information to have. And you know, if you look at this was just photos from the Eyre Peninsula back when that plastic sylvestris resistance broke down. You know, 2002-3 we had no disease. 2003 it broke down. 2 years later, after all the farmers stopped growing this, it started coming alive again. And so really what we want to do is be in a situation where we can grow it, switch to a different resistance group and then switch back to it, etc., rather than going the other way where we ended up inadvertently selecting cultivars with the same resistance genes. If you go to the Blackleg Management Guide, which is on the GRDC website, lists all the cultivars, all the Blackleg ratings. If the seed companies pay for it, they also get a Blackleg rating plus a fungicide, and then we get all the resistance groups, so you can work out what genes are in each individual cultivar. And if you do need to switch cultivars, you can make sure that you're switching to something different. So that's really the crown canker perspective of the story before we get to fungicides, but I just wanted to quickly touch onto upper canopy blackleg infection, and I'm sure you've all seen it by now. This is sort of, first saw it in 2010, and probably since 2015, it's become quite a major concern. So it's this flower infection and stem and branch infection. What we've seen or learned over the last few years is that a lot of that flower infection, that can be quite confronting when you see it, but it doesn't always cause yield loss. What we're really looking for in yield loss now is this type of issue, where we're cracking the plants open and seeing all this blackened pith inside, so it's really much more like the crown canker stage. We're not just looking for the leaf lesion infection, we want to cut the plants open and look for the crown infection, because that's the signal that's causing disease. And this is the same. Like the individual flower infection or a few little lesions like this may not actually be that significant from a yield perspective. It's when we get this internal infection in the stems and branches that it's a problem. And we're also very confident now that you can get small infections like this, and again, it may not cause much yield loss at all, but it can progress from this to this quite quickly, where you do get that yield loss. One of the reasons, or the main reasons, upper canopy has become a problem is this is a survey we took of growers from what they were doing in the year 2018 compared to the year 2000. We've got the months of the year here, and then when the blackleg spores are being released during the season. So you can see here, blackleg spore release begins in May and then goes through to September. And 20 years ago, we were sowing in that May period, and then we were flowering mid to late August going into September. And what we've seen over the last 20 years is through improved technology, we can now sow more in that April period. But what has happened is our flowering window has moved into this late July, early August period, which coincides with this spore release and cool, wet conditions for the spores to germinate, etc. So really what we've done is we've moved our reproductive part of the plant into the period of the year which is very conducive for disease. And we've simply seen new infection from stubble, new spores from stubble directly infecting our flower stems and branches and causing us a problem. Conversely, there's been a really good news story. Our seedlings, because we're sowing our plants early, we're sowing them obviously earlier in the season, but also into warmer conditions. and we're establishing through that vulnerable seeding stage a lot quicker. So we're actually seeing a lot less crown canker, I think, these days. We're avoiding that disease. So instead of sowing here and then going straight into the spore production, we're sowing here and we're getting a gap before the disease really becomes severe, and we're avoiding seeding infection, and we're getting less disease of crown canker. But unfortunately, we've pushed our flowering window into that infection period, so we're getting it later in the year. So we win on one and lose on the other, unfortunately. The other thing I think is having an influence on upper canopy is the way we handle our stubble. With satellite guidance, etc. now, we're leaving a lot of stubble completely intact. And what we've noticed is that when stubble lies down on the ground, it stays moist, all the reproduction occurs, all the spores are getting released. When the stubble stays standing, I've put them two side by side there, that is getting delayed in the growing season. So with our lying down stubble, we're seeing more spores released early in the growing season, which are going to cause more seedling infection and crown canker. With our standing stubble, we're seeing spores released later in the growing season, which are going to contribute more to flower infection than upper canopy. So we've really seen a changed farming system has changed the way the disease is attacking our plants. The farming systems now mean we're less vulnerable to crown canker and more vulnerable to this upper canopy blackleg. So now going from a control perspective, so this is, sorry, half my screen's covered here. I'm just trying to remember what I talked about here. So from upper canopy blackleg control perspective, we can see very, very major yield losses. We've measured yield losses up to 40%. We regularly measure 20%, but at the same time, we also regularly measure no yield loss at all. So it is very variable. We know that major gene resistance, when effective, will completely control upper canopy, so you get no upper canopy at all. We know the real risk is the early flowering, the earlier sow and the earlier flowering causes a much higher risk. And that's also something I often think one of the solutions to upper canopy will be the breeders producing cultivars, which you can sow early, but then stay vegetative during the winter and still only come into flower in late August. So there'll be solutions around it in that perspective, I think. So it's really about knowing what time, not so much about what time you sow, but what time you commence flowering in the growing season. And we also now know that the obviously the fungicides are very effective at controlling upper canopy. The other interesting thing is we've said right from the start it was only major gene resistance which could control upper canopy. The quantitative resistance had no effect at all. One of the things we're doing with GRDC now is trying to develop phenotyping systems to help the breeders actually select for and breed for upper canopy resistance. And we found in a lot of situations that was actually untrue. The quantitative resistance was doing exactly the same as what it was doing at the crown canker. It was actually, so here's two cultivars, the two group A cultivars, one's an MS cultivar, one's an MR. And you can see the MS cultivar, you've got really strong infection right up through the pith. And with the MR, you've got a lot less infection. So this doesn't seem to be consistent for all cultivars, but this effect is certainly there. And it might be a reason why you do or don't get responses to foliar fungicides, but I think it's quite exciting. And I think in future, it gives us the opportunity to actually try and develop a blackleg rating system specifically for upper canopy. So hopefully at some point you can be confident to actually not go and spray, to know that you have got good upper canopy resistance in your cultivars. So close.
Just bear with us for a couple of minutes. We'll just see if we, I might get actually, there's a question here while we're waiting. Pete, we've got an Andrew Smith that's asked why you're not recommending applying Marava Star post 20% flowering. Is this due to residual in grain or efficacy or on disease?
Yeah, okay. The response to that is that at the moment, Europe do not have MRLs or, they haven't determined the MRLs for pitiflumethafen. And so what we're looking at is to make sure that we do have nil detectable grain or nil residue, I should say, in grain that's being exported. We're quite comfortable with application at the 20% flowering stage. So that's where it sits at this point in time. The other reason as well is particularly given the extended residual we're seeing from Marava Star, where we are seeing residual out to 6 weeks residual. So we don't see any need to be increasing the the flowering time. So we are quite comfortable internally at out to 20% flowering. So bearing in mind too, if you've got upper canopy blackleg coming in at that semi-elongation through to early flowering, strategically application of Marcroft Star will work in quite nicely then.
Nelson's just not sure why, but his computer's shut down. He's just getting it up and going again. But in the meantime, I thought I could just quickly run through some of these questions. I don't know if anyone else has actually answered them or not. The first one, why are we not recommending applying Marcroft Star post 20% flowering? Is it due to residues in grain efficacy on disease? I think it's registered up to 50%. If someone can tell me if I'm wrong, but one of the things pardon? Whoa, we're getting a massive echo now. We've got both computers going, I think. I'll just shut this down. Excellent. We will? I assume so.
I don't hear any more echo, Steve, so we're good to go.
Hello, can you hear me?
Yes, Steve, got you loud and clear. Thank you.
Okay, great. I've just bear with me for 30 more seconds. Lost our PowerPoint now. Anyway, while Adrian's doing that, I'll talk about the application timing with the upper canopy. So one of the things I've certainly noticed working with upper canopy is that the first disease you see, the flower infection, those first little lesions turning up on your stems and branches, they're actually quite superficial until the pathogen then grows into the vascular tissue where it causes the yield loss. So what I've seen is that the fungicide applications are actually really good at giving you a bit of curative effect. Um, 30% to 50% bloom is a good timing, because at that point you're starting to see that infection. When you put it on, you can then kill those initial infections, and then you get the 2 to 3 weeks of protection from the fungicide, and then any infection that actually occurs after that, again, is superficial, and it's actually then too late for the disease to get into the vascular tissue to cause the yield loss. So by the time it's done that, the grain's already in the bin. So the 30% timing also is very indicative of a lot of work which was done with sclerotinia, and that was about as late as you can go and still get really good penetration of the fungicide down into the canopy. When you get too many flowers, you get a lot of blockage and you don't get the good penetration. So the more I see the fungicide use, the more I think just, hold fire if you can. I know it's incredibly weather dependent, and a lot of agronomists will tell me that. But if you can hold fire to at least that, I think the 30% bloom stage, it just gives you, by the time the pathogen gets going after the fungicide has run out, is then too late for it to cause any yield problems. And I think we're about to go live again. We're just scrolling through the presentation now. Get back to where we were. I think you're going way too far. Back. 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 That's where we were. Fantastic. Is anyone still there? Hopefully we don't have any more technical issues. That was a good one, losing the whole lot. Anyway, so I think that's where we were talking about, resistance. Okay, so now we're going to go into the last part of the talk, and this is just about decision making with fungicides, like I said, which is a lot of people were talking about from now on. First thing I want to say with fungicides is that the yield responses you get from fungicides are really, really variable. We know that. So therefore, we also know that when we do our fungicide work, it's very, very reliable at controlling disease, but disease doesn't always mean yield loss. And really, as an agronomist, I guess that's really where you've got to actually be skilled in knowing and being able to identify, is this a situation which is high risk? and likely to cause yield loss, and therefore I'll get a really good response to my fungicide? Or is it a situation where I've got disease and the fungicide will control the disease, but the disease was never that severe that it was going to cause a yield loss in the first place? And that's really what I want to talk to you about. So, scrap that one. The first point of call, I think, is the Blackleg CM app. So in this app, we developed this with DPER, Department of Ag in Western Australia. What you do is you go through, it's an economic app, so you put in potential yield, grain, price of the grain, etc., and it'll predict what your economic return is. You then go and put in all the parameters which will affect how much Blackleg you're going to go. You'll put in your cultivar. The app will already know what Blackleg rating is and resistance group and whatever. And put in distance to 1-year-old stubble, standing or lying stubble, all those parameters. It'll then predict how much yield loss you're going to get. And we've been verifying this app for the last few years, and it's actually coming out really quite accurate. It's up to the agronomists to know what the potential yield is. And obviously during the growing season, you can keep updating that once you get a bit of information on how the seed is progressing. But what the app will do then is predict how much yield loss you're going to get from Blackleg. And the real nice thing with the app then is just enables you to go and play with it, to actually go through, okay, what happens if I put a fungicide on? How much yield loss am I going to get? What happens if I put a Marcroft Star on? Because we've updated it with all the new chemistries. How much yield loss, etc., am I going to get? What about if I say, okay, Bonito's lost too much resistance, I'm going to switch to the newest, latest hybrid. How much yield loss am I going to get? So it enables you just to go through those parameters and play with the app. So I think that's a really, really good place to start. And then once you've done that, and then you've sown your crop and you're into the growing season, you can use this app, but then you can also use your skill to actually get out into the crop and do some scouting and then make those decisions of whether you're going to put a fungicide on or not. So from a crown canker perspective, are you going to come back with an earlier foliar? As I said before, we're talking about if you've sown the crop really early, the crop's got established before the winter period, before now, really, when we're seeing the Blackleg lesions turning up now. But if your crop's already at the 5th or 6th leaf stage now, this time of the year, it's really grown through that vulnerable seedling stage. You don't have to worry about coming back with a foliar fungicide. But if your crop looks like this, where your cotyledons have already been infected, you can see the pathogen there is already getting into the crown. This, to me, is really, really high risk. And this is where you would really benefit from a foliar fungicide. But if your oldest leaves, like I said, are at the 5th or 6th leaf stage, and you've got some Blackleg coming in, but it is probably very low risk, and the potential of getting a yield return in that situation is quite low. So the first thing is getting out to the paddock. Have I got leaf lesions or not? And what is the growth stage of the crop? Is it a small seedling, high risk? Is it 5-6 leaf stage, low risk? The next thing is actually to then relate that to the Blackleg rating of your cultivar. So again, here, if you've got no disease, means there's no Blackleg present, or it means you've got an effective major gene, no disease. That's a really easy decision. But most of our crops don't look like that. Most of our crops in our good canola growing regions with high rainfall are going to look like this. And then we have to make that decision. Is this amount of disease worth protecting with a fungicide or not? And in this situation, if you've got a good if your Blackleg rating is sound, an MR, RMR or whatever, you probably won't get a lot of crown canker. If it's an MS, you definitely will. So this level of disease is really about knowing your region, cutting crops each year and getting a pretty good understanding of what cultivars are doing what in your region. And then when you come back, you can make a much better, more informed decision on whether this crop will or won't respond to a foliar fungicide. And the other thing is, I think, which is crucial with scouting is just being aware when this situation occurs. There were photos I took in 2016 when we had that really wet year, and it really didn't matter what situation, everyone was going to get a benefit from a fungicide. So it's good to know that if that situation's occurring in your paddock as well. So once we've got through that early decision making, and I said, like, these days, I think there's a lot lower need for that early foliar because we're sowing our crops that much earlier. The key one then comes into this upper canopy infection decision making. And this is much harder because straight away we don't know what the Blackleg rating of our cultivar is. The other thing is, as Pete was talking, we'll get sclerotinia control. These fungicides will control other diseases such as powdery mildew and a bit of Alternaria and all sorts of things. So we'll get control of other diseases. But when we come to making a decision for Blackleg, we don't have an app, we don't have a management guide. We're trying to develop them at the moment, but currently we don't have them. So it makes it difficult. We don't really know what the resistance of your cultivar is. If it does have effective major gene and you've got no leaf lesions or whatever, that's an easy decision, but you have got leaf lesions, we don't know whether that cultivar is going to be really susceptible or not. So there's a lot of unknowns here, unfortunately. What we do know is that if you've got leaf lesions occurring, that you don't have major gene, so that puts you into the high risk situation. We know that if your commencement of flowering is early in the season, certainly in July and early August, then that puts you in a high risk situation as well. So this is really what I'm talking about at that stage. If you're getting out into your crops, you know, in the middle of July and your crop's elongating, they're coming into bud and you can see new leaf lesions occurring, that shows that the disease is very active in your crop, and you know then that you're in a high risk situation. If you then come back at that, let's say you're going to spray at 30% bloom and you come back and you're starting to see flower infection of that, again, you can be much more confident in putting a fungicide on that it's going to cause yield loss. And certainly if you start seeing stems and branch lesions occurring at that time, again, you can be more confident. Whereas if you're not seeing this type of infection, there's a lower probability of getting a yield return. The other thing I think with our upper canopy is to come back at the end of the year and really have a look for this, this pith damage. So one of the things we've seen, I get a lot of agronomists who say that 30% bloom spray made me an absolute fortune. And I get a lot of agronomists who say completely wasted time, we didn't see any yield return. And that's really where we're trying to work at the moment to give people much more reliable advice. But one of the key things I think you can do is come back at the end of the year and look for these symptoms. So rather than looking for just flower infection and then spraying a crop and saying, well, I didn't get any disease, actually looking and seeing, did that flower infection actually turn into pith damage and then cause yield loss? So the methods I'm using these days is to get my secateurs and just start cutting stems and branches and looking for this type of response. It's really easy to see, really easy data to collect. Again, if you want to go to the effort, you can slice the stems and branches and look for that pith damage. Or you don't even have to do any of that. You can just walk through your crop and look for what I'm calling now darkened branches. So we're getting 100% correlation between these darkened branches and this pith damage. So the plants look fine, and then right on windrowing time, all of a sudden the branch will just start collapsing, and that's where we're really. So we've collected these branches with darkened piths compared to the ones which have stayed green. And when we collect them, this is where all the yield loss is actually occurring in your crops. So rather than just looking for flowers, get out at the end of the year and then look for this type of damage. And that is actually where I'm going to leave the talk, because Sean's already talked about the fungicide resistance. So hopefully that gives you an idea. If anyone's got any questions during the growing season or whatever, give us a call. But the key things at the moment, the decision making in the next few weeks is get out, look at your seedlings, have you got leaf lesions, and is it the cotyledons, first few leaves which are really badly infected or not, because that's I guess the key selection, and know the blackleg rating of your cultivar. And I will leave it there. And sorry about the technical issues. I haven't had that one quite so catastrophic before.
No worries. Right, thanks, Steve. Look, unfortunately, we've chewed into a bit of time with that technical glitch, but we do have a few questions. Yeah, we've covered one. I think we covered one while you were down there. So we've covered off Andrew Smith's question. Danny Vivek's got a question, the comment on stubble, standing and lying. Is that example reflective across Victoria or in drier areas as well as in higher rainfall areas, if that makes sense? So I think, you know, is it reflective in both high rainfall areas and low rainfalls with standing stubbles and lying down?
Look, from a pathogen perspective, yes, it is, but it's disease is very moisture driven, so more rain, more disease issues. So look, it's much more of an issue, I think, in our high rainfall zones where you're growing your canola right next to last year's canola and you've got more disease issues. Whereas if you're in the Mallee, in a drier region, and you're not growing canola that often, it's much less of an issue, just because of the disease is much less of an issue. So I think if in the Mallee, the disease pressure is never that high, and you can probably control it much easier. You know, you can control it with a good blackleg rating, for instance, and then not have to worry about these sorts of things. So doesn't mean you shouldn't go out and monitor your crops and get a good idea so that you know for future years whether it is or not. But look, I think the risk is much lower in the lower rainfall environment.
Sorry, yeah, no worries, thanks, Steve. Sorry, Sub, we're going to bloody skip over you because you're internal. So go on to Philip Gardner. In general, how is the severity quantitatively rated between blackleg at early cotyledon stage compared to at flowering stage? Does this assist assessment of cost when to apply fungicide?
Yeah, the seedling stage is always going to be the most severe because it can kill the plant and you lose everything. However, it's totally different because at the seedling stage, we've got really good blackleg resistance. So if you get a cultivar that's overcome at the seedling stage, or you're growing like an MS cultivar, and then you suddenly have a really good wet year, then you're going to lose a lot of yield, and your fungicides are absolutely paramount. But most growers aren't in that situation. Most growers have got a good resistant cultivar, and they've got Saltro on the seed or whatever, or flutriafol on the fertilizer, so they're going to get through that seedling stage. The big issue we have with upper canopy is that we don't understand the genetics of resistance. So we're sort of flying blind. We're making that call. If the disease is bad at the flowering stage, we're going to get lots of yield loss if we've got no genetic resistance. Which is why we see these really big fungicide responses at that stage, simply because we don't know the genetic resistance. But at the same stage, we now know that some cultivars probably do have genetic resistance, which makes it really hard for an agronomist. So you've got two crops, they look exactly the same. You put fungicide on one, you get a big yield response, put fungicide on the other and you don't. But unfortunately at this stage, we can't give you a lot of advice on that genetic resistance. We hope to in the future.
Right, I've still got number 5. So before we get kicked off, so we might loop back around to Andrew Southerden's question. If you have good genetics with blackleg resistance, should you protect it with good chemistry and good cultural practice?
Yeah, that's a good question. Anything you can do to reduce the disease severity is a positive thing for protecting genetics. So the thing with genetics is that if they're effective throughout the entire life of the plant, so you're putting the pathogen, if you like, through a bottleneck. So the more disease, the more spores, and it's just a numbers game. If you've got 1 million spores landing on your plant and you've got a mutation rate of 1 in a million, you'll get a mutant that will overcome that resistance. If you've got 100,000 spores and the mutation rate's still 1 in a million, you've got a 1 in 10 chance of overcoming that resistance. So anything that you can do to reduce the frequencies of spores landing on your plant will help protect your crop. And we see this occurring right across Australia. We see, we know that the home of losing resistance is the Eyre Peninsula, where we're growing plants under the highest disease pressure. And when we grow plants like here in Horsham, we don't see that same generation of loss of resistance. So it is an absolute numbers game with spores. It's just straight genetics. Whether we can protect that with a fungicide or not remains to be seen. But I think there's probably good evidence that, yes, we can. We use all the tools in the kit, and less disease, less recombination, fewer spores, etc. We can have an impact. The other thing is, which I haven't talked about, is blackleg. Once the lesions form on the plants and you get those little black dots on the leaves, that's the actual asexual stage of the disease. So if you have a mutant that causes a lesion, it can then reproduce itself within the crop, which means it's overcoming your resistance. Whereas if you have that mutant land on your plant and it's causing a lesion, and then you come along with a fungicide and kill it, it obviously then doesn't get the chance to reproduce within your crop. So it's all those things which I think can have an influence.
Right, we've got another quick question from Phil. So, Gardner, if less than 500 metres from the previous canola crop, will blackleg encroach on new crop from the edges most adjacent, or is spread from previous crop, I presume?
Yeah, previous stubble. So the primary infection is always from the stubble.
From the previous stubble, yeah.
Yeah, that's right. So it's simply the further you get away from that stubble, the fewer spores you have. And look, when I did my PhD on this, we measured reducing disease from 0 to 500 metres, and then once we got past that 500 metres, we couldn't actually measure any further reduction in disease. So it's like, I think there's a fog of spores right across the whole region, and then as you're coming closer to the stubble, you get more and more and more. But you'll never completely avoid it. But yeah, it's that. And certainly, the first 100 or 200 metres is even a lot more severe than 300 or 400 metres away. You do see quite localized effect.
Yeah, righto. We better wind it up, Steve. Sorry to cut you off, but we can talk all day. So thank you again for joining us. Thank you again for joining us, everyone. We'll send out a quick webinar survey today to you today, just to get some feedback, should I say, on how we did and potentially what we might be able to do in the future. So, and we'll also send out a link of the webinar recording today. So hopefully you got some more good information out of this, and yeah, we appreciate everyone joining us, and thanks for joining us. Sorry about the little technical hitch. Nelson all owes us a beer, making sure his computer, but he, it wasn't working. Something was going wrong with it. But anyway, thanks very much, and yeah, thanks to you all the talkers, and hope you can join us next time. And we'll make sure Nelson's computer's working properly.
Well done, Steve.
Righto, thank you very much. Yeah, good work. Thank you.